Good morning. This is the second video or the second part of the final revision video, and we will start by the intestinal barrier. First of all, you might be asked to describe the intestinal barrier. The intestinal barrier is actually nothing but the intestinal mucosa or the wall of the small intestine, and it consists essentially of the epithelium. The IECs are the intestinal epithelial cells. So the intestinal epithelial cells are the cells that line the wall of the small intestine. These intestinal epithelial cells are characterized by three characteristics. Number one, they form a monolayer, meaning that they are one layer. They are not several layers of cells, one above the other, and this is important because the wall has to be thin in order to allow absorption. The second characteristic of the intestinal epithelial cells is that they have tight junctions in between. They have protein filaments, a very complex structure of protein filaments called the desmosomes, and the desmosomes attach the intestinal epithelial together to prevent the intestinal epithelial cells together to prevent leakage in between the cells. And the third characteristic we studied is the one we studied in the digestive system, and it is the presence of those microvilli or the brush border that increases those folds towards the lumen of the small intestine, increase the rate of absorption. And then we have a mucus layer, and that mucus layer lies towards the intestinal lumen or the cavity of the small intestine. And on the other hand, we have the lamina propria. Lamina means layer, and it is called propria because it consists of connective tissue proper, a very delicate connective tissue that holds the intestinal epithelial cells together, and they rest upon it. And on the side of the lamina propria, we have plenty of immune cells. We have non-specific immune cells that can act against any pathogen, for example, the dendritic cells and the macrophage, and we have specific immune cells that act against specific pathogens, like the B cells that produce antibodies against the pathogens, and the T cells that release chemicals in order to kill cells where the pathogen has been introduced. The mucus layer acts as a sieve-like layer. It doesn't look like a sieve, but the molecules of mucin, the protein that forms mucus is made of a protein called mucin. So in between the molecules of mucin, there are spaces that regulate the passage of substances. That's why we call it, we say that, or we describe it as being like a sieve-like layer, in addition to two important molecules, the AMPs or antimicrobial peptides, and the SIGA or the surface immunoglobulin A. The antimicrobial peptides and the surface immunoglobulin A are protein molecules that are embedded in the mucin layer or the mucus layer, and that act more and more to control what passes, because they will kill every suspicious pathogenic molecule. The AMPs, as their name indicates, are antimicrobial, meaning that they can kill microbes. And the immunoglobulin is an antibody that is there to help attack the pathogen. And immunoglobulin A in particular is called a surface immunoglobulin because it is always present on the surfaces of the mucous membranes, on the mucus layer. And that's why we call it surface immunoglobulin A. Now, the intestinal barrier is described as being semi-permeable. I might ask you to justify. So what types of questions actually can be encountered here? Number one, I might ask you about the structure function, adaptation of the intestinal barrier, how the structure of the intestinal wall is well adapted to carry out its function, its semi-permeable function. So you're just going to describe what I've just said, meaning that you're gonna say that the intestinal epithelial cells are a monolayer to facilitate diffusion and absorption. They have desmosomes in between to prevent leakage. On their outer surface, they have immune cells to prevent the passage of pathogens. On the inner surface, they have the inner surface, which is towards the lumen. They have mucus to protect them against alkalis. And they also have those AMPs and surface immunoglobulin G, immunoglobulin A, to prevent pathogen entry. So the intestinal barrier is, the second question is, why do we justify the intestinal barrier is semi-permeable? Why do we say that it is semi-permeable? This is because it allows the passage of useful beneficial molecules, like, for example, all those simple, small, soluble molecules that are present in the lumen of the small intestine, but they restrict or prevent the pathogen of the pathogen entry. This is another kind of question, but the third type of question might be a very simple question, which is just describe the structure of the intestinal barrier, and it would be the easiest question to answer. And by the way, I would like to emphasize that your answers should be precise answers, meaning that you shouldn't write too many details, and at the same time, they should be very relevant answers related to the. One of the important functions of the intestinal barrier, in addition to being semi-permeable, allowing the uptake of essential nutrients and preventing pathogens, is immune sensing. And immune sensing means that it detects the presence, it senses or detects the presence of pathogenic organisms and gets rid of them. And immune sensing, of course, is a function of all these immune cells, the non-specific cells, and the specific cells. The second concept, or the next concept we will move on to, is how are you first exposed to microorganisms? So I might ask you, for example, a fetus inside the uterus of his mom is usually sterile, means that it does not get contact to any kind of pathogens. So how are microbial flora, how are friendly flora actually developed on his skin and in his gut? So, and by the way, there was like a kind of mistake in the previous video when I said that you have to mention the genome. You have to mention the genome when you define a microbiome, but you don't have to mention the genome or the gene pool when you just define friendly flora. So the question might be, how do we acquire friendly flora, or how does a sterile newborn acquire the friendly flora? Now, first of all, it is through the normal vaginal delivery, because the vagina of the adult female, which is the mom in this case, is full of friendly flora coating the inner mucosa of the vagina. So when the membranes around the fetus rupture and there is this gush of fluid to push the fetus passes through from the uterus to the vagina, and through the vaginal wall, it acquires a lot of friendly flora that find their home or find their way first on the skin of the fetus, and then they stabilize themselves there. So this is the first way we acquire skin flora. The second way we acquire skin flora is from the environment. The environment, the air around us, and everything is full of bacteria. Some of them are pathogenic, but they'll not stabilize. They will not be able to stabilize themselves on the skin because of the presence of the friendly flora, and the friendly ones will stabilize. So first from the vaginal wall, second from the environment, and third from handling. Of course, handling by the medical doctors is not a source of bacteria usually because they wear sterile gloves, but handling by the parents and the members of the family. These are the three ways to provide skin flora to the newborn. But there are other ways we get the gut flora, and the principal way we get the intestinal useful flora is through breastfeeding. So breastfeeding is mainly related to the gut flora because on the skin of the maternal breast and the nipples skin, there are friendly flora. And these friendly flora, while the baby is suckling, will be transferred to his mouth and then down his gut. So breastfeeding is a good way of acquiring gut flora. But there is something that's not written here on the slide, but that will come on later slides or show on later slides, which is why is breastfeeding preferred to bottle feeding? Breastfeeding is preferred to bottle feeding because of two reasons. The first reason is the one we have just mentioned. Suckling allows you to acquire skin flora from the breast of the mom. But the second thing is that breast milk itself contains growth hormone or a growth factor for the bacteria. It contains a growth factor, for the bacteria. And that while the baby is suckling milk, this growth factor will be moved to the inside of the gut of the baby. What's the importance of the growth factor in the milk of the mom? The importance of the growth factor in the milk of the mom is to help the very useful and very important bifidobacteria in the gut of the mom to flourish, to what do we mean by flourish? To reproduce and then to grow. So this was the answer to justify breastfeeding is preferred to bottle feeding. But here there is another related question, which is justify that normal vaginal delivery is preferred to C-sections. By C-sections, we mean cesarean sections. So, of course, this is because of what we have just said, that the normal vaginal delivery allows the newborn to acquire friendly flora from the inner mucosa or the inner lining of the vagina. I might ask you to define symbiosis or symbiotic relationships, or I might ask you, what does symbiosis refer to? Symbiosis refers to any relationship between any two organisms that belong to two different species. Of course, symbiosis is further subdivided into three types of relationships: commensalism, mutualism, and parasitism. Now, what does each of these mean or refer to? Commensalism in general refers to a relationship, a symbiotic. So every time you're asked to define these three subclasses, which are commensalism, mutualism, and parasitism, you have to start by it is a symbiotic relationship. So commensalism is a symbiotic relationship between two organisms of two different species, where one of them gets benefit, like food and shelter, and the other one neither harms nor benefits. Concerning the relationship between gut flora, for example, or even skin-friendly flora and human beings, this does not apply. So I might ask you, justify there is no true commensalism in the relationship between friendly flora and human beings. This is because actually friendly flora take home and shelter and food from human beings, but in exchange, they provide benefits. they're not neutral, they're not harmful. So they provide benefits. So if they get something and give something, it can't be commensalism, it would be mutualism. So the actual relationship between bacteria and the friendly flora, actually, whether they are in the gut or in the skin, and human beings, is actually mutualism. What kind of benefits do they provide? We've just mentioned before in the previous video two important benefits, which are producing vitamins like vitamin B and vitamin K, and providing fatty acids that kill pathogens. And here we are adding two benefits, which are, number one, providing local immunity. Local immunity meaning immunity in the space where they are present. How do they do this? For example, gut flora will provide gut immunity. Skin flora will provide skin immunity. This is what we mean by local immunity. So how do they provide this kind of local immunity? By weakly stimulating the immune system. There is a sort of a deal between the friendly flora and the immune system, meaning that whenever we see pathogens, we're going to stimulate you weakly in order to attack those pathogens. Of course, it is weak stimulation because the antigens of the bacteria are weak antigens. An antigen is a substance that is capable of stimulating the immune system. So whenever they see a pathogen, they stimulate the immune system, and the immune system will attack those pathogens. But meanwhile, they have taught the immune system not to attack them. So letter A on this slide is one way how friendly flora can kill pathogens. Letter B is also another way how they can kill pathogens. So A and B represent an answer to two types of questions. You might give this answer, those two answers, A and B, when I ask you about the benefits of friendly flora, or when I ask you describe how friendly flora kill pathogens. So the second way is competing with pathogens, meaning that when they reproduce, when the friendly flora that represent the majority reproduce, they take up space, and therefore they don't leave enough space for the pathogens to be colonized. But we have also studied a third way in the previous video, which is producing fatty acids, because these organic acids, like fatty acids, will kill the pathogenic bacteria. So up till now, we know three ways of killing pathogens. And we will still learn more about the ways friendly flora kill the pathogens. So you have to compile all these pieces of information that are related to fighting pathogens. The second symbiotic relationship is mutualism. And in mutualism, there is a kind of a mutual exchange or a mutual relationship between friendly flora, between two organisms of different species. And the best example is the relationship between friendly flora and human beings, because as we said before, if I ask you, there is no true commensalism in the relationship between friendly flora and humans, but it is mutualism. Justify, it is mutualism because there is exchange of benefits. How? Friendly flora take home and shelter, and in exchange, they do so and so and so. For example, they kill pathogens, they produce vitamins, and you can give details like they occupy space, they secrete chemicals, and so on. Parasitism is a relation, again, if you are asked about, as I've just said, if you are asked about the definition of parasitism, you have to start by, it is a symbiotic relationship. Every time you're asked to define commensalism or mutualism or parasitism, or if you are asked such a question, what do they refer to? You have to start by, it is a symbiotic relationship. In parasitism, it is a symbiotic relationship where a living organism takes benefit, and in exchange, it gives harm. And the best description, of course, for these are the pathogenic flora or the parasitic worms like bilharziasis, like ascariasis, and or ascaris, and tinea with an A. Now, your microbiome is, of course, unique. It's like a fingerprint. And here it is important, as I mentioned before, to mention the genome. So the microbiome, you have to add here, there is a missing thing, which is, it is the gene pool or the genome of all the microorganisms in a particular environment. I've just added it. So you might start by genetic pool, or you might say, and their genetic pool. What are the factors that make so I might ask you directly, what are the factors that make your microbiome unique? Or I might give you the statement, every individual's microbiome is unique, and I might ask you to justify. So the first cause is genes. Everyone inherits specific genes from their families. And the second thing is the way you were delivered. Of course, it would make a difference, because if you are normally delivered through the vagina, then you will take a specific microbiome from your mom. The way you were fed, because if you were breastfed, then you will acquire a specific microbiome from the skin of the breast of your mom. And then age. Age is important in determining your microbiome, not because as you get older, the number of friendly flora diminishes, the diversity diminishes. So the bacteria becomes less diverse and the beneficial microbes decrease. So there are two changes. Number one, it becomes less diversified, and number two, the total number decreases. So if the total number of friendly flora in particular decreases, then those chemical signals that were released, as we have watched in the video at the beginning of the semester, there are chemical signals that are released and that are transferred from the small intestine across the blood to the brain to stimulate the brain and give health to the brain and improve your mood and improve your behavior. This kind of relationship is called the gut-brain axis. So the gut-brain axis is that highway that connects the gut to the brain, and it is actually the blood vessels mainly. So if the friendly flora diminish, then the gut-brain axis diminishes, and this is one of the interpretations or the explanations why Alzheimer's disease occurs. Alzheimer's disease is a disease in which brain cells degenerate, brain cells deteriorate. Their function declines and diminishes over time, and this is because of this diminished connection between the gut and the brain. So taking care of your diet and of your gut microbiome is important to preserve your brain. We also found, scientists found differences in the microbiome between the different genders, and it wasn't related to the diet, meaning that if they receive the same diet, they still have different microbiomes, and this might be due to hormonal differences. Males have male hormones, the androgens like testosterone and androsterone, whereas females have female hormones like estrogen and progesterone. Also, stress harms the healthy ratio, 85 friendly to 15 pathogenic flora. So when you're under continuous stress, then you will have a disturbed ratio of friendly to pathogenic flora. And stress is present all over your life. For example, weaning is stressful for an infant or a young child. Teeth eruption, when you lose your milk teeth and the permanent teeth start to erupt. Puberty, of course, for both males and females, is a stressful age. Menopause or cessation of menstruation, all this alters the composition of the normal flora, partly because of hormonal changes and chemical changes in the body. Diet is very important to determine your flora. As we have said before, you are what you eat. So if you eat healthy, you will be healthy. What kind of healthy food improves your gut flora? Mainly plant fibers, as we have said before. Justify why are plant fibers important? Because for your health, because plant fibers cannot be digested by human beings. They're digested or micro-digested and broken down by bacteria to get a small amount of energy. And therefore, when you take plenty of plant fibers, you're just helping the friendly flora to increase in number. And this kind of plant fibers is what we call the prebiotics. On the other hand, if you take refined grains like white flour, for example, grains without the outer coat, wheat, for example, without the outer coat, refined sugars, white sugar, and animal products, all these are food for harmful bacteria. And that's why when a person has cancer, we ask the person to restrict sugar, proteins, and white flour or none or refined grains, from his diet. And lastly, we talk about medications. Of course, there are certain supplements, natural supplements that are important for improving your gut flora, like the probiotics. Probiotics are pills that contains friendly flora, but there are other medications like immunosuppressive drugs, like antibiotics, that kill the good flora with the pathogenic flora, and therefore they disturbed your ratio. Now, if we have a friendly flora or a group of friendly flora, can they change behavior and become pathogenic? Yes, they can. What are the reasons that I might ask you to justify this statement that friendly flora can turn pathogenic? There are several important causes for this. Number one, immunocompromisation. When you have a very weak immune system, whether congenital immunocompromisation, meaning that you're born with it. Congenital means that you're born with it, or acquired, meaning that you were born normal, but for a reason or another, for example, due to medication or due to an acquired disease like AIDS, for example, you became immunocompromised. I might also ask you about the acquired, but let's first say the big titles. So the big titles are immunocompromisation, hormonal changes, and damage to the skin or the mucous membranes. First of all, we defined immunocompromisation. It is an extreme weakness of the immune system. And we said that immunocompromisation is of two types, congenital and acquired. And now the next question would be, what are the causes of acquired immunocompromisation? Acquired immunocompromisation can be due to chemotherapy or to radiation therapy, and both are used in cancer. Chemotherapy causes immunocompromisation because it kills the white blood cells that are produced in the bone marrow. So if you are giving chemotherapy, it circulates in blood, it will kill most of your white blood cells, whether the ones inside the bone or the ones in the peripheral blood, and therefore it causes immunocompromisation. Radiation therapy will also do the same thing if it is radiation on bones. Radiation therapy does not circulate in blood. It's like rays, high energy rays that are directed towards a particular organ. Number two, the hormonal changes. And of course, we have seen that at puberty, there is a change in the behavior of the friendly flora P. acnes. Instead of being useful, it turns pathogenic. And this is all due to a surge in androgenic hormones, with all the consequences that happen in the sebaceous gland to make it turn into a swelling, an inflamed swelling on the skin. And we will be discussing this in detail later on. And the third factor is damage, to skin or mucous membranes, because we have said several times that as long as your skin is intact or mucous membranes lining, for example, your mouth cavities or your stomach or your small intestine are intact, they are not breached, then this is a good mechanical barrier to prevent microbes from penetration. On this slide, we're going to discuss the different shapes of bacteria. We cannot talk a lot about bacteria without trying to visualize them, or without your visual memory having an idea about what they look like. So we have three main shapes of bacteria. We have the spherical bacteria, or the cocci. Cocci is the plural and coccus is the singular. And we have the rod-shaped bacteria, or the bacilli. And then finally, we have the spirilla, or the spiral bacteria. So these are the basic morphological shapes of bacteria. When we say morphology, the title of the slide is Morphology of Bacteria. Morph means shape. So morphology is the study of the shape of bacteria. And inside every group, we might have subclasses. For example, in the group of cocci, it might be a monococcus or a diplococcus. So if I ever ask you to define a diplococcus would be spherical bacteria arranged in pairs. And then we have streptococci, and streptococci are spherical bacteria arranged in chains. And then we have the sarcina. First, the tetrads. We have tetrads. They are spherical bacteria arranged in groups of 4. And then we have the sarcina, which is like spherical bacteria that are arranged in groups of 8. So when you have 8 spherical bacteria attached together, 8 spherical bacteria attached together, then it is sarcina. And then we have staphylococci, a group of spherical bacteria that are arranged in a grape-like shape. And then we move on to the slide here is the same as the explanation I've just given. And then we talk about the skin microbiome. The skin is one of the largest organs in the body, and it is considered an organ because it consists of multiple tissues. And the total surface area of the skin, of course, varies from one person to another, and the average surface area of the skin is 2 square meters. As we have said before, the skin microbiome is acquired from the inner lining of the vagina of the mom. We say vagina, but we say vaginal. The adjective is vaginal. But there are certain areas of the skin that are most abundant with friendly flora. What are the areas of the skin that are most abundant with friendly, what are the skin areas where the microbes are mostly abundant? We have the axilla, the groin, and the areas between the toes. Justify, this is because these areas are warm and they are moist, either because of perspiration, which is very slight sweating, or sweating. So warmth and moisture are 2 important reasons why they are there, in addition to the fact that it is good shelter or good home for the bacteria. As we said before, once the pathogen has, once the bacteria or the microorganism has settled on the skin, this means that it is mostly a friendly flora. And as we said before, the relationship is mutualistic. Can you give examples on this slide? We are giving examples of friendly flora having a mutualistic relationship with human beings. We can give the example of staphylococci, or we can give the example of P. acnes. These 2 bacteria are essentially friendly, but as we said before, under certain conditions like puberty or the presence of a skin wound or just an abrasion, they might turn pathogenic. Why do we say that they are mutualistic? Because they take home and shelter and give benefits. What are the benefits provided? We are repeating ourselves. So you can choose any of these benefits and write them on the test if it is required. They occupy space, meaning that the more they are, the more space they occupy, and the fewer pathogens that are allowed to be present in the space. Number two, they produce organic acids like fatty acids that inhibit the growth of fungal organisms like the unicellular fungus candida. And number three, as we said before, they stimulate the local immune system. Local here means where they are present. So here it is the skin immune system. But as we said before, at puberty, P. acnes might change their behavior. Why? Because they get trapped. And when they where do they get trapped? They get trapped in the sebaceous glands. What happens when they get trapped? They become pathogenic. What happens when they become pathogenic? They grow rapidly. They release chemicals that they didn't release before, or they weren't used to release before. And these chemicals, they weren't used to releasing those chemicals before, and those chemicals cause inflammation of the sebaceous gland and the hair follicle, because the sebaceous gland is related to the hair follicle. When the sebaceous gland gets inflamed and when the associated hair follicle gets inflamed, this is what we call acne vulgaris. So what is acne vulgaris? Acne vulgaris is a chronic inflammation of the skin associated with those swellings on the surface of the skin that are full of sebum, or a sebaceous substance or a fatty substance, and they might not be inflamed at the beginning, but at the end of the day, they become inflamed, they become swollen, they become warm, they become painful, and they, of course, become red. So these are the symptoms and signs of acne vulgaris. So I might ask you, why is acne vulgaris considered a chronic inflammation? Of course, this applies to any disease that is chronic. Whenever you're asked this question, you have to mention three entities: a gradual onset, a prolonged course, and a gradual onset, a prolonged course, and very mild symptoms and signs, or almost mild symptoms and signs. In this drawing, we can see the pathogenesis of acne vulgaris. What is the pathogenesis of acne vulgaris? Means how acne vulgaris develops. First of all, there is an androgenic surge, meaning that there is a rise in androgen hormone. This causes changes in the sebaceous gland. What are these changes? Number one, epidermal proliferation. There is a rapid rate of cell division here. So there are cells that are shed out, and those cells accumulate here and block the sebaceous gland. But is it the only reason why sebaceous glands are blocked at puberty? No. The second reason is the changes that occur in sebum. Sebum increases in quantity, increases in thickness, increases in stickiness, and therefore this is a second factor in addition to epidermal proliferation that blocks the gland. When the gland duct is blocked, because it is an exocrine gland that has a duct, what happens is that the secretion accumulates inside the gland. When the secretion accumulates inside the gland becomes swollen and forms a comedone. Comedone is the swollen sebaceous gland, but it's not yet inflamed. After a while, the P. acnes get mad. They become pathogenic. They reproduce rapidly. They cause inflammation, and the comedone gets inflamed. So we can see not only a swelling on the surface of the skin, which is the comedone, but we can see redness, and it is extremely painful and hot. Sometimes we have, of course, all the changes that occur in the sebaceous gland occur in the hair follicle. What is the hair follicle? The hair follicle is the pocket of skin that surrounds the hair and protects it. And sometimes on the surface of the skin here, we can see a black dot that is called the comedone. And this indicates that the condition is severe, that it is a severe inflammation. And those black dots are nothing but dead cells, and it is a black head. We call it a black head from dead tissues. So what is acne vulgaris? It is a chronic skin disease. Chronic skin disease characterized by what? By blockage first, and then by inflammation of the sebaceous glands and the hair follicles that are associated with them. Are there factors that help this? Yes. First of all, genetic factors, meaning that all these changes we've talked about due to the androgens become more probable to occur if you are genetically predisposed. So genetic factors are an important factor in determining the occurrence of acne vulgaris, and it's familial. It runs in families not only because of the genes, but because of another factor, which is the culture and the diet. Are there specific food types that help in this? Yes, there are specific food types that help the occurrence of acne vulgaris or even increase the severity of symptoms of acne vulgaris. Number one, the nature of the skin. Of course, if you have oily skin, this means that you have more sebaceous glands and that you are more liable to get acne vulgaris. If you eat nuts, if you eat chocolate, if you eat dairy products, all these are factors that are bad for acne vulgaris and increase the severity of acne vulgaris. The hormonal factors have been mentioned on the previous slide, so we're not going to repeat them. And of course, the presence of friendly flora like P. acnes that was essentially friendly and that turned pathogenic. How do we treat acne vulgaris? We have to do deep cleansing of the ducts of the sebaceous glands and the openings or the outlets of the hair follicles. And the best cleanser is salicylic acid. Salicylic acid is aspirin, actually, but salicylic acid has when it is in a solution, it has a very important function, and that important function is to remove all these debris. It washes away all these dead cells, so it removes the comedones and it removes the cells that accumulated from epidermal proliferation, so it opens the duct of the gland, helps release of the accumulated fat. If salicylic acid alone is not enough or is not effective, then we can use antibiotics, but we use antibiotics very rationally, as we said before, and only if there is inflammation. If it's just comedones, if it's just swellings that are non-inflamed, we do not use antibiotics. And then now we move to the nasal cavity. We're gonna discuss every cavity in the human body and discuss the microbiome inside it. The nasal cavity microbiome consists essentially of the grape-like spherical bacteria that are called Staphylococci. So it's a common finding. However, not all people have or harbor Staphylococci in their nasal cavity. If you harbor Staphylococci in your nasal cavity, but do not show symptoms and signs, then, let's first discuss if you harbor Staphylococci in your nasal cavity, then you are a nasal carrier, just by harboring them in your nasal cavity. So if I ever ask you, what is a nasal carrier? A nasal carrier is a person who harbors Staphylococci in their nasal cavity. But if I ask you, what is an asymptomatic nasal carrier? An asymptomatic, here, there is an added word, which is asymptomatic, an adjective. So if I ever ask you to define or to say, what does this refer to? What this refers to, which is an asymptomatic nasal carrier, you would say that it is, again, a person who harbors Staphylococci or friendly flora. We say Staphylococci because it is the most commonly present one. It is a person who harbors Staphylococci in their nasal cavity but do not show any symptoms and signs. Another question is, justify staphylococci are considered potential pathogens. Yes, they are potential pathogens because they are not essentially pathogenic, but they can turn pathogenic under certain conditions, for example, when the immunity of the patient is lowered. Another question, that this slide is rich in questions. Another related question is, why are asymptomatic nasal carriers considered a risk to the community? Why are they dangerous? Because the answer is because they can transfer such staphylococci to others. They can be infectious to others, and other people might catch, for example, might inhale the staphylococci and get the disease. So what's wrong with this? What is wrong with this is that those nasal carriers are asymptomatic. They do not show any symptoms and signs. And the risk comes from here. They do not show any symptoms and signs. Therefore, they themselves do not know that they are diseased. And therefore, people around them do not know that they are diseased. And therefore, although they are asymptomatic, people do not take the necessary precautions, and they themselves do not take the necessary precautions to keep apart from other people, and therefore the transmission of infection is easy. Another risk is, but it's not a risk to the community, it is a risk to the carrier itself, is that if that carrier coughs or sneezes, the nasal discharge or saliva, the nasal discharge that is released in the form of small droplets, droplet infection from the nasal cavity, or saliva upon coughing that is also released as small droplets, will spread in air and might fall on his own skin. So if his own skin is abraded or injured, so the person might, or those nasal carriers might infect themselves, and this is what we call self-injection or auto-inoculation. Auto means self and inoculation means injection. Why do we say injection? Because it is as if they injected the microbe into their skin. So they might harm themselves and they might harm other people. How do nasal carriers spread infection? There are two essential ways how nasal carriers spread infection. The first way is droplet infection, and the second way is direct contact. Droplet infection. What is droplet infection? Droplet infection is a way of spreading infection when the person sneezes or coughs and droplets of nasal discharge or droplets of saliva are released into air and can be inhaled by other persons. But droplet infection itself is, or droplets, the droplets themselves can be transferred via three ways. The first one is airborne droplet infection, when those droplets of saliva or mucus are transferred via air to another person and that person inhales the droplet itself. The second way of transmitting droplets is dust-borne droplet infection. In such a way, the droplets fall onto dust, and then the dust particles dry out, or the discharge, the droplets dry out onto dust because of air and because of the heat or the high temperature of the weather. And in such a case, if there is wind and that dust is carried again by air, another person might inhale it. And the third way droplets can be transferred is through food. So we have food-borne droplet infection. For example, if you cough or sneeze and those droplets fall onto food, on a plate, for example, onto an apple or onto whatever kind of food, rice, pasta, whatever, and the person ingests those droplets of infection. But in such a way, if it is food-borne droplet infection, most probably it will not cause a respiratory tract infection, but it might cause, if it's, for example, a bacterium that might cause vomiting and diarrhea, it might cause gastrointestinal diseases. The second way nasal carriers can spread infection is direct contact. Again, for example, if the person coughs or sneezes into his hands, then the hands are full of staphylococci, and when this dries out, the person might forget that his hands are not clean. So it's a very bad habit or a very bad routine to cough or sneeze into the hands. So you now have contaminated skin of your palms. So if you shake hands with others, you are transmitting the infection to them. If you touch a countertop or toys or books and magazines, for example, with your contaminated hand, then now the bacteria are present on the contaminated object. And for example, the third example of direct contact is when you bite an apple, for example, or drink from a cup, and then another person comes and eats from the same place where you bit the apple. And now we are going to move to the conjunctival microbiome. The microbiome in the conjunctiva, of course, first of all, what is the conjunctiva? The conjunctiva is a thin, very thin, delicate, transparent membrane or tissue that covers the sclera of the eye and meanwhile coats the upper lid and the lower lid from inside. And it starts at the edge of the colored part of the eye. It starts at the edge of the cornea. The cornea is the convex transparent thing in front of the pupil of the eye, and therefore it does not cover the cornea. The conjunctiva is very important because it is protective to the eye. And I might ask you to justify why is infection of the eye or of the conjunctiva infrequent. The infection of the eye or the conjunctiva is infrequent because there is mechanical protection and chemical protection. Or in other words, there is a mechanical barrier and a chemical barrier. The mechanical barrier is in the form of a thin film of tears, which we call the liquifilm. Why does this cause a mechanical barrier? Because it mechanically prevents the bacteria from penetrating the conjunctiva. This is number one. Number two, microbes and dust will stick to that liquifilm. But this is not all about the mechanical protection. There is also blinking, because you have trapped the bacteria on your liquifilm, but they're still there. But blinking with your eyelids will constantly wash away this liquifilm with all the bacteria and pathogens in it. And as you see here, those are the lacrimal glands on the outer aspect of the eye. They secrete tears, and then the surplus of tears passes through a lacrimal means related to the tears, to the nose. We don't feel the tears that are removed. We don't feel the liquifilm as it passes through our nose constantly because it is a very thin liquifilm and a very small amount of tears. But of course, if you cry, then there will be a lot of tears, and then a lot of tears will pass through the nasal duct, and you will have a runny nose. The chemical barrier is the presence of the enzyme of a chemical substance, an enzyme called lysozyme enzyme, inside the tears. And it's not only tears that contain the lysozyme enzyme and that do this chemical protection. Every fluid in the body, like saliva, for example, contains lysozyme. Sweat contains lysozyme. So if lysozyme is present in all these bodily secretions, especially the surface secretions, then lysozyme is important. Yes, lysozyme is very important because lysozyme is an enzyme. So it catalyzes a certain chemical reaction. What is that chemical reaction? It is a chemical reaction of hydrolysis or breaking down of the cell wall of bacteria. You know that all bacteria are prokaryotes, and all prokaryotes have a cell wall made of peptidoglycan, proteins and carbohydrates. So if lysozyme enzyme breaks down this, then the bacteria no more have a bacterial cell wall, and therefore they are destroyed and they are killed. This is the chemical barrier. I will stop here and I'll be uploading the video, the second part, on Google Classroom, and then we will be continuing, inshallah, in other videos.